she's giving us the signal to go ahead. It's good. Okay, we're going to go ahead and get started. Thank you all for joining us today for the debrief session over this, or the debrief over the session, the Human Cell Atlas: Transforming Biology and Healthcare. My name is Callie Jones. I'm a student journalist and a PhD student studying cancer biology at the Mayo Clinic. And so we just came from a really wonderful session talking about this Human Cell Atlas initiative. And so I want to first let I'm fortunate enough to be joined by all of the speakers at that session, and I'd love to start by allowing them to introduce themselves and give us a couple of words about your role in the Human Cell Atlas initiative.
I can go first. I'm Aviv Regev. I'm a core member and the chair of the faculty and the director of the Clarence Cell Observatory at the Broad Institute. I'm a professor of biology at MIT, and I'm an investigator of the Howard Hughes Medical Institute. And in the context of the Human Cell Atlas, I'm actually one of the two founding co-chairs, together with my great colleague Sarah Teichmann, who's at the Sanger Center.
My name's Casa Maya. I work at the Sanger Institute in the UK with Sarah Teichmann, who's of course the second co-founder, and my role at the Sanger is really to lead some of the HCA projects within the institute.
My name is Shannon Hughes. I'm a program director at the National Cancer Institute, part of the NIH. And actually, my role in the HCA is from the outside. I've subscribed to their Slack channel and others in order to keep abreast of what the HCA is doing, but my main role is to support the Atlas building initiatives at the NIH, such as the Human Tumor Atlas Network or HubMap, which is another initiative that's building atlases of normal human organs.
We can be collaborative with our microphones. Oh, this one's louder. Okay. Okay, so the stated mission of the Human Cell Atlas Initiative, which many of you mentioned during your talks, is to create a comprehensive reference map of all human cells, which are the fundamental units of life, as a basis for both understanding human health and diagnosing, monitoring, and treating disease. That is a lot. All put into one mission statement. And you each, I thought was very interesting, had a different analogy, a different metaphor that you used to explain it. Aviv was talking about a periodic table of cells. Kirsten was talking about Google Maps. And Shannon, this was probably my favorite, the spiral-bound Rand McNally Atlas in the back of your car that you used on family road trips. Which I identified quite strongly with as the child of the family who was the one using the spiral-bound Rand McNally Atlas. But so I kind of wanted to, either using those analogies or not using those analogies, because that mission statement, the Human Cell Atlas, is such a complex animal. I kind of wanted to hear each of you, in your own words, explain, what the Human Cell Atlas is and what it means from your own perspective.
So I will focus on what it would be rather than what it is. And what it is includes all the activities that we do and so on. And I think the critical thing is, what do we want to see at the end of it? And I think what we want to see at the end of it, regardless of favorite metaphors, is kind of the same thing. It's the ability to know, first of all, which cells we have and all of their characteristics. They have many different characteristics. And to be able to understand what healthy looks like, and in what way do we deviate from it in disease. The Human Cell Atlas itself focuses on the healthy, that's why it's a reference, but the disease efforts give us this notion of where we go beyond. And in this, we want to capture all the properties of cells, all their aspects or facets. We want to be able to say the different kinds of cells that are there, the different programs that they run, the different neighbors that they might have, the places in their body in which they sit in terms of, the tissue histology they sit in, the place in our anatomy that they sit in. And so we have this comprehensive view of all of these properties. The reason that I allude to a periodic table is that a periodic table, and so I hope would be a Human Cell Atlas, is more than just a description of things that we know. It is actually a model. And so Mendeleev had a periodic table before he actually measured all of the elements. He could predict that there are elements there that we didn't yet see. And I think one of the things that is at least an aspiration that we would get is that we would be able to push beyond everything that we've measured and to be able to say, even if we didn't see a cell there, there surely has to be one.
So that was already a very comprehensive view. I think I would like to just emphasize the concept of cell atlas, because as it says in the mission statement, the cell is the fundamental unit of life. That is really where we translate the genetic information into function. And for me, that is one of the really exciting sort of conceptual ideas of the Human Cell Atlas, that you begin to understand the regulatory mechanisms and then also how cells influence each other. But it is that at that cellular level that we're trying to define it, and I think that's where genetic regulation occurs, and I think that's hugely interesting.
And maybe I'll pick up from that, because when we think about Kirsten speaking from the genetics to the cellular level, I think there's a whole nother aspect that we think about in disease, which is how the cells manifest into disease, and are there emergent properties that we find through cell-cell interactions, or understanding the transcriptional programs within each cell and how that affects the cell next to them. So thinking about a multi-scale view of this, so trying to think about spatial as well as all of the clinical information, like I showed in my talk, that can be tagged to understanding how cell interactions manifest into their normal homeostasis or disease.
Thank you, guys. So this question is more to Aviv, who is one of the co-chairs of the project. Can you give us a little bit of history on how this project, how this initiative more like came to be? Where did the initial idea come from? How did it develop?
This is a fun question to answer. And so I'll start with my own personal perspective of history, because it's the one I have firsthand, and then I'll put it in broader context. So many of the things that I showed today, even though these were, the latest and greatest that we've done maybe, they have already become apparent, at least to me, by about 2014. By that point, we've had examples where we went into a piece of tissue and we got the cell types out, where we could, as a community, recover developmental processes, things that I haven't even shown today, like figuring out where the cells actually sit in three-dimensional space, questions that had to do with disease, so we could see it would be valuable in diseases like cancer. And we had those droplets that were running in that image that also existed by then. And so we had the sense that if we had a big atlas, we would be able to give these amazing answers, and we had the technology to show that we could build something as big as that. And so in 2014, I actually got an email from a program officer at NAGRI called Adam Felsenfeld, that Shannon knows well. And he said that they're running one of these strategic conferences or meetings that they have. It was a couple of days in some, nondescript hotel in the DC area. And he asked me whether I wanted to give something called a challenge talk about anything I wanted, about something that we could do which would be meaningful if I had 5 years and $50 million. And I was like, absolutely, I'm going to give a talk. And so I pulled together a bunch of slides. Actually, my first slide, the one that shows the human and the cells and they make into boxes, that is from that talk. I still love it. And the name of the talk was the Human Cell Atlas. And I stood there for I got 5 minutes, and I said, look, we should do it. We could get all these things. We have these droplets running around. They weren't published yet. And we should have a Human Cell Atlas. And it became a little bit of a personal mission. I had this small deck of 10 slides that would stick to every talk that I would give. And for a couple of years, I just went around, and every talk I said, we should build a Human Cell Atlas. And started writing these white papers to try to convince people that it was a feasible thing. But what you absolutely have to understand in academic research and in open research is that ideas sprout in kind of this ever-going way. And so other colleagues were thinking along these lines. Stan Linnerson, who studies the brain and is also one of the pioneers in single-cell genomics, he was busy, working on the brain, a tissue that we work on as well. And it was kind of resonating for him as well. Sarah Teichmann and her colleagues had a center, whose name I'm not sure I will quote correctly, but they had a center focused on single-cell genomics, like we had the Klarman Cell Observatory at the Broad. So, they started feeling, and people, I think, saw me with those slides, so they knew I was a kindred spirit. And so in early 2016, I got an email from Sarah saying, I know you're interested in doing that thing, and we're interested in doing something like that. Should we do something? And we agreed that we should have a meeting. That's a very, scientist thing to do. And so we agreed that we would try and bring the community together. And we convinced Katrina Gold and Michael Dunn from the Wellcome Trust to allow us to hold the meeting at the Trust. They agreed to give us a room. It had 93 seats. And that's the maximum number of people that we could bring to our meeting. And they helped with a tiny bit of budget, like for the food. People had to pay for their own way for travel. And together with Katrina and Michael, we started working on the idea of what would a meeting like this mean. And it's not just a meeting, it's what the thing would mean. The meeting is just a form that follows function. So we started brainstorming this together. What would it actually take? All these presentations and white papers and discussions and centers are not the same as an initiative. Who should come to a meeting like this? How to convince them to come, because it's for a thing that doesn't exist. We had nothing to offer, not even travel money. And we worked through that. We had a pre-planning meeting in August 2016. Katrina had a spreadsheet that showed all the declined invitations, like all the people who didn't want to come. We made a bit of an effort to convince some important people that they should really come. It would be worth their while. And we all went to London. I still have a photo like that. We took of all of the name tags sitting in the front, in the table in the entrance. And it was a very special moment for both Sarah and I, because we felt like this thing, it could be a reality if only we convinced everyone. And then we walked into the room, right? Everyone was lined up to give their talks. And everyone was already convinced. We weren't like doing a hard sell at all. And over the lunch break, we set up the organizing committee of the Human Cell Atlas. And that was the launch, not of HCA itself, but of its planning process, which was broad and open and community engaging, because it wasn't for any one of us to say, this is how we'll do it. It was for all of us to do it together. And so it was. And in 2017, October 2017, a year later, as we promised, we launched a formal atlas.
Also say a little bit of how this has grown through some funding agencies as well. So in 2010, the NIH Common Fund program funded the single cell analysis program, or the SCAP program. And that's really where we developed a lot of the technologies that are paramount to the Human Cell Atlas and these other atlas building initiatives. So, both within the grassroots effort that Aviv describes, and then within a variety of different funding agencies, there was a realization that that the best use of those particular tools was in building things like cell atlases right now. So, I think that a couple different things happened organically and now require a lot of coordination on everyone's part, and I think everyone's dove in to make sure that that happens.
I'll add one thing, that just like this has happened for the Healthy Atlas, so have efforts like the Human Tumor Atlas Network that grew out of the Blue Ribbon Panel for the Cancer Moonshot. Scientists coming together, exchanging their ideas openly, brainstorming how can we do something, and then just going after it.
So I wasn't there at the beginning of the Cell Atlas, but I think one of the things that you guys did brilliantly is really emphasize this open science and open data sharing, because I think that has made a massive difference to allowing the growth of the community that could then coalesce around this, in a way, quite simple idea, something that people can really relate to, just map all human cells. But it wouldn't have been possible if people held on to their own little research and not been open and shared and encouraged others to join.
That is actually a perfect point. Talking about one of the things that you all emphasized very strongly during the course of this is that this is a very collaborative initiative, and that it is a global collaborative initiative, and that it could only be possible if everybody comes at it from their own things and all contributes and works together. So I guess to drive home that point a little bit more, why is it so important for this to be a global initiative?
I can start. I think it has two separate values that both require it. The first is that we're actually committed to equity and diversity in research in the scientist side. And the reason for that is that we all know, and this is well backed by research, that more diverse communities are more creative, have a broader range of ideas, and end up doing better in any problem that you want to solve, surely in a problem that requires the degree of creativity that science requires. The second and just as important reason is that we have learned from, decades of genomics that we're actually diverse. We're genetically diverse to begin with. And so if we do not actually have the breadth of reach that is needed in order to look at human diversity, how can we claim to have a human cell atlas? It's not a particular human, it's humans in general. But I also want to emphasize that you could achieve genetic diversity without geographic diversity, but that's not enough for cells. Because unlike my genome, which is reasonably static, as I was in Seattle and come back, and the sun shines and the sun sets, and I fall asleep and I wake up, my cells are actually not static at all. They are changing. And so we also need to see this diversity of environments in which humans live in order to really get a good spectrum of how cells are. And so for all of these reasons, this is one of those cases where there's only one correct answer, and that's to do things internationally and globally.
And I think the current coronavirus outbreak just really, really highlights that, none of our societies work in isolation anymore. We are all incredibly highly connected, and a disease that emerges wherever in the world can and will spread around the globe. And we really have to work as a community and get everybody involved, everybody to be open about what diseases are emerging, how we can tackle it. And actually the Human Cell Atlas here again is a beautiful example of how by generating this reference atlas, Aviv had a lovely slide, we can now look at, once somebody finds out what is the possible receptor for the new coronavirus, we can immediately go back to our reference atlas and say, well, where is it expressed? Does this help us to explain how the virus is spreading?
From a funder's perspective, sometimes it's difficult to fund science outside of the country in which the funds are coming from, right? So we have, led by Richard Conroy, who's at the NIH, part of the HubMed program, really started to put together an international funders group that meets periodically, who's funding different parts of the Human Cell Atlas and the different NIH Atlas efforts to make sure that we're coordinating our efforts and that people aren't building tools and infrastructure that will work against one another, right? Because one of the things that we aspire to, it's very, very difficult, but what we aspire to is having data sets that will be interoperable, or you can use them across different programs. That's a whole nother session and a whole nother conversation, but we recognize that, and from a funder's perspective are trying to take steps to facilitating that more easily.
So with a global initiative and a collaboration of this scale comes a lot of promise and a lot of hope, and you all shared some really interesting pieces of data that have come from this initiative so far, and so I'm thinking to close this session, I would like to hear what is your favorite thing, your favorite insight that has come from the Human Cell Atlas project so far. Give you a second to think. I know that is a weighty question. Sure.
We'll give them a chance to think. So again, I'm going to step back and talk about an NIH program, but one of my favorite things was something that I talked about, the patient interaction that the Kidney Precision Medicine program is really acting at the forefront of with regards to involving patient participants within their creating their research protocols. They have a seat at the table. So I think that's one of my favorite outcomes so far of some of the NIH programs. We're still waiting on a lot of scientific outcomes, but just at the get-go, making sure that we have the best interests of patients in mind.
So for me, I think it's always easiest to get excited about things where you know a lot of the details. So for me, some of the work that's been done at the maternal-fetal interface has actually been the most exciting, because also this is an area where it's absolutely critical that we know exactly not only what are the single cells, but what is their genetics. Are they from mum or are they from the baby? And being able to use the single cell biology and start to understand the interactions that allow the fetus, the conceptus, to start to develop and to be nurtured versus preventing a rejection of this foreign body within the mother's body has been a really exciting area.
So it's very difficult to choose, okay? So I'm just going to choose the one about which I'm the most emotional. I think they're all fantastic, or many of them are just unbelievably fantastic. And that one has to do with the intersection of cancer and health biology, which is in very devastating pediatric tumors of the brain called DIPG. They're a form of very aggressive gliomas where there is nothing today. There is zero therapy. There is nothing to offer the patient who will be a child of a relatively young age or their parents. There is only sadness. And my colleagues Mario Suva and Mariella Philbin, who both treat patients like this and their families and are research scientists, have used these single-cell genomics approaches to ask, but what are these tumors even made of? Which is something we did not understand well. They're incredibly rare. You only get tiny biopsies with about 1,000 cells in them taken from the children's brains for diagnostic purposes. They're not operable. In doing these studies, they have totally transformed our understanding of the disease. They have built a whole model for what is actually going on. We understand the cell of origin now, what goes wrong in brain development that ends up 6, 7, 8 years later in a young child as causing this disease. They understood the cells that are likely the root cause and that we should be targeting therapeutically. And they can now use these techniques, not just to compare it to the healthy brain and understand how it's different and so on, but to start screening for drugs. So emotionally, every time I run into Mariella Philbin, who, you know, for her day job treats the patients and in the night studies their cells, I tell her, I'm privileged that what we've done is able to help you in your research. And she always says, in the sunniest possible way, and we will find a cure. And for that, I'm emotional.
Thank you guys so much for those, those impressive stories, and thank you so much for, just the research that has been done on this project and the different ways that you've contributed. The session itself was very powerful, and thank you for agreeing to speak with us a little bit more about that. For those of you who weren't there, the session was a packed house. There's standing room only, and it was, it's clear why, this is a great initiative. So I think we're running short on time. I don't think we have time to take any questions from the 5 minutes. Okay. So are there any questions in the audience? Does anybody have anything for our speakers? Sure. Feel free.
This seems like such an obvious thing to do, and the need for it seems so obvious. How did we not already have this?
We couldn't. We didn't have the technologies. So I often tell people building a cell atlas has always been an endeavor, about 150 years old endeavor. You can take it all the way back to the 1600s when the microscope was invented and Hooke saw cells for the first time and called them cells. Biologists have been trying to characterize cells ever since, and every time they had a new technique, one of the first things that they did was turn around and try to look at cells again, try to put them in categories. I alluded in the panel discussion to Monica Hall's work going from early 1900s is really the first transformation when we had new techniques to do that. So the challenge was that we had many, many different ways to look at cells, but they weren't unifying. And it was very hard to do it for the massive numbers of cells that you would have to look at, each of them alone, separately from the others, either when they're organized in a tissue and you can identify this one versus that one, or when you dissociate them individually. Those technologies have started to emerge maybe 8 years ago. They've become robust enough that you could imagine doing it at this scale around 2014, 2015. For some of them, we're still inventing. So we've been really quick the moment that we could. That's how I would put it.
Yeah, okay.
Yeah, so...
To repeat the question, since it wasn't on a mic, she's asking for examples of the technologies that are being used that have led to this.
I'll mention briefly because I know Shannon wants to add. So one example is what we call single-cell genomics, and within it I'll say single-cell RNA-seq, where you can profile the molecular contents of individual cells, and you can do it for a lot of cells. Another category of technologies are what we call spatial transcriptomics or spatial proteomics, where you can do the same thing, look at a lot of molecules, but you do it when the cells are still inside the tissue. For example, you do it under a microscope or using other kinds of techniques.
So I think it's also important to acknowledge that pathologists have been doing this for a very long time, right? Yeah, they can look at a slide and tell you whether or not it's normal tissue or diseased tissue, but what these efforts will allow us to do is to systematically measure that and get that knowledge that's inherent in a pathologist's brain out into the community to allow more people to do data science and draw hypotheses that can be tested. So I think that that's one of the reasons why it's timely important. We are not going, the question was, are we going to put pathologists out of business? And the strong answer to that is no way.
They are our best friends and very active participants in HCA.
You have to use a mic.
Well, I think we're out of time, actually. But thank you all so much again for being here, and let's give our panelists a round of applause. Thank you guys so much.